citric acid or TCA cycle. Now the TCA cycle, again, plays a very central role in metabolism. The TCA cycle deals with what do we do with the pyruvate that we produced at the end of glycolysis. Now pyruvate, as we discussed already in previous sections, is the final product of glycolysis. Pyruvate can also be produced not only by glycolysis but also by the degradation of certain amino acids. Now once we've produced a molecule of pyruvate, it can have one of three possible fates. One is that pyruvate can be converted to glucose via gluconeogenesis. And we talked about this already when we discussed gluconeogenesis. The other thing that can happen to pyruvate is it can be completely oxidized in the citric acid cycle to produce reducing equivalents that are used in oxidative phosphorylation to produce ATP. We'll discuss that in a separate section. The other thing that can happen to pyruvate is it can be converted to lactate, and this happens in anaerobic conditions. Let's talk about what happens under aerobic conditions first. Under aerobic conditions, and when we say that we mean when there's plenty of oxygen around, under those conditions pyruvate is transported into the mitochondrial matrix where it's converted to a molecule of acetyl-CoA. And the enzymatic reaction is described as follows. Pyruvate is combined with a molecule of CoA plus a molecule of NAD+ to form acetyl-CoA plus a molecule of carbon dioxide plus NADH. This reaction is irreversible, and this is an important concept. Once acetyl-CoA is formed, it's committed to one of two fates: oxidation to carbon dioxide in the TCA cycle, or conversion to fatty acids, which we'll discuss later. You cannot convert acetyl-CoA back to pyruvate and go through gluconeogenesis. Now, the reaction by which pyruvate is converted to acetyl-CoA is catalyzed by an enzyme called pyruvate dehydrogenase. Pyruvate dehydrogenase is actually a complex of three different enzymes, referred to as E1, E2, and E3. And each of these enzymes has a different cofactor. The E1 uses thiamine pyrophosphate, E2 uses lipoic acid, and E3 uses a molecule of flavin adenine dinucleotide, or FAD. The enzyme complex is feedback regulated by levels of acetyl-CoA, NADH, and ATP. So in other words, when acetyl-CoA levels are high, there's a negative feedback regulation. Similar things happen with NADH. And ATP is a signal of positive energy balance, under which condition you would not want to turn the TCA cycle and produce more ATP. Now, each of these things stimulate phosphorylation and inactivation of the E1 subunit of pyruvate dehydrogenase. Now, once acetyl-CoA is formed, one of four things can happen to it. It can be oxidized in the TCA cycle, it can be used to form amino acids, it can be used to form ketone bodies, and it can be used to form fatty acids. And we'll describe each of these at some point. Now, under anaerobic conditions, what happens to pyruvate? Well, anaerobic conditions are conditions in which there's no oxygen. And so under anaerobic conditions, we don't want pyruvate to go into the TCA cycle because we can't do oxidative phosphorylation to consume the products of the TCA cycle. So rather than doing that, what we do is take the excess pyruvate and convert it to lactate. And this is done by an enzyme called lactate dehydrogenase. Lactate dehydrogenase takes pyruvate plus a molecule of NADH and converts it to lactate plus a molecule of NAD+. This reaction consumes the NADH produced during glycolysis. If that didn't occur, NADH would build up and glycolysis would be inhibited. Lactate is generally produced under anaerobic conditions in red blood cells, remember, which always function under anaerobic conditions, and in the muscle when it's exercising, and it's taken and transported into the circulation. It's taken up by the liver, where it's converted back into pyruvate by lactate dehydrogenase. This is a reversible reaction. And then the pyruvate is converted back to glucose via gluconeogenesis. This cycle of glycolysis and gluconeogenesis is known as the Cori cycle. So let's take the example of exercising muscle. In exercising muscle, glucose is converted to pyruvate by glycolysis. But remember, there's no oxygen around, so the pyruvate, rather than being converted to acetyl-CoA, is going to be converted to lactate by that enzyme lactate dehydrogenase. This is regulated simply by the level of substrate and product in the cell. Under anaerobic conditions, there's high concentrations of pyruvate. This activates lactate dehydrogenase, and you produce lactate. That lactate gets transported into the blood and is taken up by the liver, which converts it via lactate dehydrogenase back to pyruvate. And again, the direction that the lactate dehydrogenase enzyme goes depends upon the concentration of substrates and products. In the liver, the lactate level is much higher than the pyruvate level, and so the reaction proceeds in the reverse direction from lactate to pyruvate. That pyruvate then is converted back to glucose via gluconeogenesis, and that glucose can be released into blood, and the whole cycle repeats itself over and over again. This is the Cori cycle. Now there can be defects in this process as illustrated in this clinical vignette. A 3-month-old girl is brought to her pediatrician because she doesn't seem to be acting right. When you ask with the parents what they mean by that, the parents say that she's becoming increasingly lethargic and seems to be eating less. They also state that she doesn't seem to be very active and she's kind of floppy. She doesn't have good muscle tone. When you look at her growth charting, it shows that she's actually lost weight over the past month. She's tachypnic, but her other vital signs are normal. Her physical exam reveals poor muscle tone. She can't hold her head up, and her grasping and sucking reflexes are weak. Deep tendon reflexes are hyperreflexive, with clonus in the lower extremities. We do some laboratory tests, and you show that there's a metabolic acidosis with an elevated anion gap. So what is the disease that this patient has? Well, this is pyruvate dehydrogenase deficiency. Pyruvate dehydrogenase deficiency is a rare deficiency in one of the 3 enzymes of pyruvate dehydrogenase, typically the E1 subform. There are 3 forms of this disease. There's a severe form which causes overwhelming lactic acidosis and usually death in the neonatal period. There's a moderate form where you get a milder acidosis that causes neurologic damage due to metabolic deficits in the brain. That's because the brain relies on the TCA cycle for energy, and if pyruvate dehydrogenase isn't working, the TCA cycle can't function properly. There's also a mild form that causes episodic ataxia, which is induced by a high carbohydrate diet. Now, whether the form is severe, moderate, or mild depends upon the degree to which the pyruvate dehydrogenase activity is decreased. The E1 defect is X-linked, but affects heterozygotes as well as homozygotes. And this is an example of X-linked dominant inheritance, one of the few examples that there are. We treat this disease by giving patients a high-fat, low-carbohydrate diet. This decreases the lactate production because they're more dependent upon fat for generating energy than they are on carbohydrates. And it increases production of ketones, which is an alternative energy source for the brain. We can also give these patients high-dose thiamine, and this stimulates whatever residual pyruvate dehydrogenase activity there is. Now, in this section, we also want to consider how one other thing is metabolized, and that's alcohol. Alcohol that's consumed in the diet is usually in the form of ethanol. Ethanol is metabolized to acetate via an acetaldehyde intermediate. And this requires 2 enzymes. One, alcohol dehydrogenase, which converts ethanol to acetaldehyde, and the other, aldehyde dehydrogenase, which converts this acetaldehyde to acetate. Both of these reactions produce a molecule of NADH, and both of them require thiamine pyrophosphate as a cofactor. Now, there's an important clinical correlation when we consider this pathway. The alcohol methanol is also metabolized by the same pathway, but rather than being converted to acetaldehyde, methanol is converted by alcohol dehydrogenase to formaldehyde, and formaldehyde is very toxic. One of the sequelae of formaldehyde toxicity is blindness, and so people who consume methanol may lose their vision because of formaldehyde toxicity. The treatment of this disease is actually ethanol administration. If you give somebody ethanol, this competitively inhibits the alcohol dehydrogenase. It competes with methanol for metabolism and prevents the conversion of methanol to formaldehyde and decreases the toxicity associated with methanol poisoning. Okay, so now we've talked about a couple of different ways that we can generate acetyl-CoA. The main one being pyruvate dehydrogenase. Now, once we have acetyl-CoA, we need to put it into the TCA cycle and completely break it down to carbon dioxide to produce energy. The citric acid cycle has several different names. One is the tricarboxylic acid cycle, or TCA cycle. One is called the Krebs cycle, named after the biochemist who characterized it. These terms are all interchangeable. But whatever you call it, this is the process by which acetyl-CoA is completely oxidized to carbon dioxide. And in the process, we produce reducing equivalents in the form of NADH and FADH2, and we produce also GTP. Now, we call it a cycle because the other components, other than acetyl-CoA and carbon dioxide, are completely regenerated. This is a picture of the citric acid cycle. One of the things that's really daunting to students of biochemistry is seeing all these biochemical steps with all these enzymes and thinking, I've got to memorize all this, and it's really challenging. But again, as I've mentioned in other cycles, I want to break it down to a very simple form and only consider the most important enzymes. Now the inputs and the outputs of the TCA cycle are as follows. These are the things that aren't recycled. You have an input of acetyl-CoA, 3 molecules of NAD+, a molecule of FAD, a molecule of GDP plus inorganic phosphate, and 2 molecules of water. The outputs are the CoA from acetyl-CoA. You get 3 NADHs, 1 FADH2, and 1 GTP, and 2 molecules of carbon dioxide, which are the 2 carbons that came from the acetyl-CoA. Now the TCA cycle has to be regulated like all biochemical pathways, and we do this to ensure that the energy homeostasis is maintained. In general, the TCA cycle is activated when energy needs are high. That is, when the ADP to ATP ratio is high, or the NAD plus to NADH ratio is high, and vice versa. When energy needs are low, when the energy balance is positive, we're going to have relative inhibition of the TCA cycle. Now, regulation of the TCA cycle occurs at 3 different steps, and these are the only 3 enzymes that are important for you to memorize in the TCA cycle. The first is citrate synthase, the second is isocitrate dehydrogenase, and the third is alpha-ketoglutarate dehydrogenase. And they're shown schematically in this picture of the TCA cycle. Now, citrate synthase is responsible for catalyzing the formation of citrate from acetyl-CoA and oxaloacetate. This is the first step in the TCA cycle. Now, this is regulated by ATP. ATP is an allosteric inhibitor of citrate synthase. It actually increases the Km for acetyl-CoA. So when ATP levels are high, that signals a high energy balance in the cell, and we want to inhibit the TCA cycle. And we do that by inhibiting the first step of the TCA cycle, that is citrate synthase. Isocitrate dehydrogenase catalyzes the formation of the 5-carbon alpha-ketoglutarate from the 6-carbon molecule isocitrate. It also reduces NAD plus to NADH and generates one of the molecules of CO2 that are produced by the TCA cycle. ATP is also an allosteric inhibitor of this, so that when energy levels are high, the TCA cycle is relatively inhibited. ADP, on the other hand, is a stimulator of isocitrate dehydrogenase activity, such that when ATP levels are low, when the energy balance is negative, we stimulate the TCA cycle. Similarly, NADH inhibits the activity of isocitrate dehydrogenase by competing for the binding of NAD+. And so when NADH levels are high, again an indication of high energy balance in the cell, isocitrate dehydrogenase is inhibited. The last regulated step is that of alpha-ketoglutarate dehydrogenase. Alpha-ketoglutarate dehydrogenase takes the 5-carbon alpha-ketoglutarate and converts it to the 4-carbon molecule succinyl-CoA. It also reduces a molecule of NAD plus to NADH, and it generates the second molecule of CO2 that's produced by the TCA cycle. Alpha-ketoglutarate dehydrogenase is feedback inhibited by NADH. Again, when energy levels are high, signaled by high levels of NADH, this step will be inhibited. It's also inhibited in a negative feedback fashion by succinyl-CoA, which is its immediate downstream product. Now, some intermediates in the TCA cycle can be substrates for other biosynthetic pathways. So the TCA cycle has multiple functions, not just the production of energy, but also to serve as a go-between in different biosynthetic pathways. For example, oxaloacetate and alpha-ketoglutarate can be used to synthesize certain amino acids, and we'll discuss this more in a different section. Malate and oxaloacetate can generate intermediates for gluconeogenesis. In addition to that, succinyl-CoA and fumarate are actually products of the degradation of certain amino acids. And so the TCA cycle can sort of be an intermediary step between glycolysis and amino acid synthesis, for example, because glucose is broken down to pyruvate and then to acetyl-CoA, which enters the TCA cycle, and then some of the TCA cycle intermediates can be used to create amino acids. It also serves as an intermediary step between amino acid degradation and gluconeogenesis. So in the fasted state, we may break down proteins to form amino acids, which can enter the TCA cycle. This produces intermediates of gluconeogenesis, which can then form glucose. And so the TCA cycle then serves as a go-between between glucose and amino acid metabolism. So here's a summary of the TCA cycle. We start with a molecule of pyruvate, and we produce 3 molecules of carbon dioxide. One of those is produced by pyruvate dehydrogenase. The other 2 are produced in the TCA cycle proper. We produce 4 molecules of NADH, again, one of those by pyruvate dehydrogenase, the other 3 by the TCA cycle proper, a molecule of FADH2 and a molecule of GTP, both produced by the TCA cycle. The key regulated enzymes are pyruvate dehydrogenase, which converts pyruvate to acetyl-CoA, citrate synthase, which combines acetyl-CoA with oxaloacetate to form citrate, isocitrate dehydrogenase, which converts isocitrate to alpha-ketoglutarate, and alpha-ketoglutarate dehydrogenase, which converts alpha-ketoglutarate to succinyl-CoA. Diseases associated with this process are pyruvate dehydrogenase deficiency, as we described, and these are the important features of the TCA cycle that you need to remember. And this concludes the section on the TCA.